in spinal muscular atrophy, musculoskeletal disorder with a neurological component, and then one in oncology, each showing here patients that have been treated with these medicines that are now available in various parts of the world, I believe, including the US and Canada. So gene therapy is a science that is relevant for patients now is the main point here. So what are the opportunities of gene therapy versus maybe other drug modalities that you may be more familiar with? Some of the key properties of gene therapy is that they could lead to a durable therapeutic benefit, that the mechanism of action of these drugs often goes to the root, often genetic cause of the disease, and that they try to address an unmet need for often diseases that don't have treatment options available currently or even in prospect. So where that leaves us is that there's kind of three Venn diagrams where the opportunities lie. Probably the most prominent one right now is diseases with a genetic etiology, and as gene therapy, we can make therapeutic interventions at that root cause here. The fact that many gene therapies require only a single dose opens up a set of opportunities that you can now try to address areas of the body that you can't access frequently, but where you can make a justification for accessing them maybe surgically once. For example, in ophthalmology, the example I showed you earlier, we tend to avoid surgical interventions in the eye on a regular basis. So therefore, if you can just go in once, hopefully fix with a durable effect the disease, all of a sudden you now have opportunities to address a need there. The fact that we can deliver a protein in a sustained manner is also an area where we find these opportunities. And actually taking all of these combined leaves us that there's a real opportunity around chronic disease and gene therapies. So we're not alone in having realized this, and there's actually an entire industry that is at an inflection point over the past decade, where if you go back to, say, 2014, this is an article that was published in Forbes then, where the excitement of the field was illustrated by the type of investment that the field had seen. These dollar figures here, expressed in millions, are now almost faded by the type of deals that are taking place in the industry here, which are illustrated on the right. So we're really entering a phase of industrialization, with large players such as Roche and Novartis entering the fray, whereas here this was much more on the NUCO and the new company side. But that doesn't mean that there's still a whole trough of companies that are emerging as well on the NUCO side here. So where do I fit in? I run an academic research lab, actually a center called the Grausbeck Gene Therapy Center. We study the mechanism of how these gene therapies effectuate and how these diseases are in need of treatment via gene therapy. Our biggest emphasis is really around technology development, and we'll talk a little bit about that in one of the case studies for sure. And then we do believe that translation is necessary to keep your eye on the ball. Otherwise, you are developing these studies in isolation, really, and we want to avoid that, and therefore are also part of the translational axis. In one particular setting, we're doing that in collaboration with a center called the Ocular Genomics Institute, where we try to bring therapies for retinal blinding diseases. But in fact, many of the aspects of the center itself goes outside of the eye and looks, as you will see in those case studies, at a variety of other indications. Our main technological tool right now, mainly for reasons that it's actually highly efficacious, it's been found safe, and we can make it, is called AAV. AAV is a viral vector system called the adeno-associated viral vector. And we in particular have studied the capsid, the external surface of this particle, extensively for reasons we'll go into in depth in a bit. But what is an AAV? An AAV is really a FedEx truck. This shell here is the truck, that delivers a particular package. And in our case, that is a nucleotide sequence that we deliver appropriately to the cell that could benefit from that therapeutic intervention. What we have learned is that this FedEx drug, this AAV shell, really determines to a great effect the efficacy, the safety, and the manufacturability of this particular type of therapies. So it is a highly complex driver of all of that biology, and part of our work is really trying to elucidate and control that biology for these three main aspects. So the case studies that I promised you are three case studies that I've walked through over the past probably now 6-7 years in my lab at Harvard, one of which was really actually foundational. In around 2015, we did what's called a sponsored research agreement with an organization called Lonza. And what we really try to bring in here is a set of resources to a technology that we had that originated out of our lab and which was done via a variety of sources of funding before we engaged with Lonza, where we took the diversity of these AAVs illustrated here in this phylogeny, and we tried to design novel AAVs based on the knowledge that we had in the space. That gave us a toolbox that we felt could help us decode AAV, if you will. We tried to understand the structure-activity relationship in this space, and we did that via developing a high-throughput screening mechanism that allowed us to explore a diversity that we had generated here. And the learnings from that allowed us to now start reprogramming vectors. What is illustrated here, for example, is one of the demonstrations that we have done where we know that many of these AAVs, when they're administered systemically to an animal or a human, they're particularly efficiently trapped to the liver. And actually that creates a lot of opportunities because many of the diseases we try to target are liver diseases, or sometimes we leverage the liver as a factory for production of a therapeutic protein. But in some cases that is actually not good because the liver is not our target. And here we show an ability to actually detarget from the liver, which actually has a bunch of safety as well as potential efficacy benefits. And we see this as a cycle, as a discovery cycle, where the learnings here now can go into improved design and we can start this process all over again. So what did we do with Lonza, and who is Lonza? Lonza is probably familiar to many of you, a global manufacturing, contract manufacturing company in pharmaceuticals and biologicals. And we did a strategic partnering with them where basically the handoff was that we provided them some access to our IP, and they committed to building manufacturing processes, something that they're particularly good at. And at the same time, they allowed us to get funding for moving on towards the next generation and kind of going through the cycle and actually building to a large extent that discovery cycle that I just described. We were quite selective. We had a number of potential opportunities, but we felt that this was a relationship that was what I call here fit for purpose, because it was still very early in where we were in the research. So it was actually unclear what the real opportunities are. So having kind of a more broad exploratory partnership we felt was beneficial. We also knew that manufacturing was key in this space, and therefore partnering with a manufacturing partner was important. And we went into the collaboration, and we'll come back to that maybe at the tail end, with existing IP. And that gave us actually quite a bit of leverage at the negotiation table, not to be completely selling out, but having this more or less an equal partnership. One of the things that actually emerged out of this is that once we started doing this research, we found one of these opportunities, and one of these opportunities eventually led to a NUCO called Akouos. So let me tell you about the opportunity and about Akouos. The opportunity is really around hearing gene therapy. I mentioned earlier that the space is quite developed on the eye, and that kind of illustrates already that neurosensory disorders may have in general some opportunities. But the ear actually, for many reasons, has been locked up from really profound therapeutic opportunities. And in a large part, that is because the inner ear, the cochlea, kind of schematized here, It's locked up in the hardest bone of the human body and actually really difficult to access. But it has some benefits and some analogies to the eye. For example, the fact that we can, if we can access it, we can do a local administration of a small dose because it's a small organ. It has immune privilege. Then there are actually a variety of these genetically defined targets. There is a need because really the standard of care is limited largely to devices such as the cochlear implant or a hearing aid. Um, but there's a challenge here that there's no precedent for any drug to be delivered like this to the inner ear. And obviously, hearing is a large unmet need, if you will. So, what we had shown is that when we stacked up a technology that we had developed versus the gold standards, illustrated here by AV1, AV2, AV8, all existing technologies, we had illustrated a greater efficiency in key cells that we needed to target, the hair cells. And particularly, actually, we demonstrated that these outer hair cells, these nicely structured row of cells here in these 3 layers, we were able to access really quite efficiently throughout the cochlea. And what we had shown in an animal model is that if we leverage this technology for a particular genetic disease, we could make otherwise deaf mice now gain hearing almost to a whisper. So really profound low sound levels that we could restore hearing function to. So that led to this company called Akuos, which we ended up setting up with a number of venture partners. Eventually, even got an additional round of funding, and actually we're fortunate enough to launch this on NASDAQ last year. But what is, I think, even more important than the money part is that we really did this with a key set of experts in the area, not just my technology, but we needed surgical expertise and we needed a variety of pieces of the puzzle to come together. Importantly, also the business experience. So we found a champion for this technology that really ended up building out the business and leading it to the success that I talked about. We ended up licensing a part of the technology to this company. And now this company is looking at actually being in the clinic next year. So a third case study after Lonza and Akuos was something very unique. I've been very much concerned about ultra rare diseases. Ultra rare diseases are diseases that are so limited in prevalence or incidence that it's very difficult to come up with a commercial model to make these ultimately into drugs. Notwithstanding the fact that many gene therapies in various settings have shown really great promise that they could intervene in these ultra rare diseases. So what we wanted to do is create a path for ultra rare diseases and gene therapy where there currently is none, at least not on the commercial side. So we got together with a number of stakeholders to build this company that really is based on the premise that in the eye, where there are many of these ultra rare disorders, and where we actually have one approved drug that was developed actually by one of my mentors when I was training, Gene Bennett. This drug was approved in 2017 by a company called Spark Therapeutics, and really is in and of itself an ultra rare form of inherited retinal degeneration. But we had realized that this is a one-off. This is only going to happen once. This was a proof of concept. It was actually the first in vivo gene therapy that was approved by the FDA. But the next targets, the appetite from the commercial world was only to go into larger diseases. So this would not repeat itself, notwithstanding the fact that there's actually a whole range of these diseases out there. So there are indeed over 200 diseases, and the challenges are really that it still is very expensive for each of these to be developed, and that it's very difficult to develop these even at an increasingly higher set of pricings that takes place in this industry. So we're kind of in a logjam, if you will, that while there's real opportunity and real need, there's many reasons that prevent these from being developed. So Odilia really tried to be a non-profit biotech. We set something up that tried to do many things that normally biotech would do, but what we tried to do is bridge kind of these preclinical findings, which we feel are available, and bring them to clinical trials, at which point there may be more traction for them. And at least we would move these sets of targets forwards. We ended up partnering these not with venture capital because they sought too much of a return of investment and they felt that we couldn't stay close to the mission, or we felt that. We did this with a patient organization, with an academic organization, and with some passionate scientific founders. The number of dollars that are available to this are much less, but we are able to at least sustain and grow this mission over time. The last one other case here is a company that I started called Affinia Therapeutics. Affinia Therapeutics really builds on this decoding of AAV and redesigning of these AAVs via a rational design pathway. What basically the main problem set is that we had seen is that many of the AAVs that are around now that are used to build into drugs are really technologies that are 20 years old, and they're called naturally occurring AAVs. And actually, many of the designs that people have done over the years have failed in really being clinically adopted. That being said, there's a general realization that we need more tools. In fact, in the clinic right now, even though there's this initial success in gene therapy after actually trying for so long and failing quite often, but there's this initial success, but that starts to level off right now. And it's really hungry for, we believe, a technological revolution, or at least innovation. So what we set up is we kind of shaped the proposition that we then later brought to venture capital to build a company that has a technology platform that wants to develop products, but that also has a big emphasis on manufacturing. And really the real unique differentiating feature is that we wanted to understand AAV and decode it towards ultimately controlling its pharmacology better through rational design. The pathway of our funding is illustrated here. We were fortunate to get indeed an incredible group of people around the table to build out this larger vision, brought in some money in a limited way that allowed us to kind of nucleate the company in a variety of ways. And then ultimately we were able to partner as well as bring in more capital to now be in what's called, in my view, an industrialization phase. We're trying to build an academic set of ideas and early concepts, but now we're trying to industrialize those. And then something that I didn't fully anticipate is a company called Albumunity. This is actually case study number 5. This was, if you will, not necessarily maybe an unwanted child, although I've actually enjoyed myself. This is something that is COVID related. It's a small project in my laboratory that emerged into snowballed really into a much larger program that is trying to develop a single dose room temperature stable vaccine on the technology that we're so familiar with. So what it really is not unlike many of the vaccines that you all have heard about and are now being approved, a gene-based vaccine. We decided to do this with a number of parties around the table that had, because we needed to go so fast, available infrastructure rather than build it out from the start, like, for example, how we did it with Affinia. We were fortunate to bring in partners like Novartis that could help us manufacturing our vaccine. And we're still very much engaged by stringing together all these partners and continue to develop this unique proposition. The main reason why we still are compelled to keep on doing this in light of some excellent vaccines that are emerging is the fact that we believe that the benefit of a single dose and the room temperature stability really allows to get a vaccine across the globe to cover particularly that last mile, the last mile for an effective vaccine to get into people's arms. And we believe that this technology has a unique ability to do this. So I'll close off here by a few lessons learned in kind of all of these case studies that are just summarized here in just very short kind of maybe pieces of advice, or at least things that I've taken away from this. The first one, possibly the most important one, that life is short. So if you are compelled to do anything like this, you have to understand that it takes an incredible amount of energy to do this successfully, so that if you don't have the passion to do this, or if some of the pieces aren't there, you may want to reconsider doing this. And I don't want to be discouraging here, but I think the realization that this is not necessarily a game is important here, and that it does take an incredible amount of energy that does take away from other things that you're doing in life. I think, secondly, very importantly, in all settings, you have to have substance to compel yourself, to compel others, to have a stance at a negotiation table. You have to have something that's real, or at least you have to be able to fully understand the limitations of what you have. I generally believe that in the end, things that you don't fully believe in will fall through the cracks. The most important thing, I would argue, from all the things that are important, is that even if everything else is in place, you can only do this when you have the right set of expertise around the table, but almost equally importantly, the right type of people that mesh with you, that you can work with, or that can work well with each other. So really the people component is essential here. This is a general lesson that I do in my lab as well as in business. I tend to try to fail fast and fail often. Certainly in biology, very few things work out. So you have to celebrate the things that do. But it is a statistical process to some extent that you have to try out a number of things before you get there. And the failing fast is important because you don't want to wait years to understand that something that you've invested in is not going to work. So to set up the right set of experiments and be partnered with people that are kind of into that approach as well. From a business and also legal perspective, two pieces here. I think learning throughout the trajectory is critical. And it's critical because mainly you want to understand how people look at you. So you want to understand what the other side of the table thinks, how they think, and how they look at you. And that often requires you to understand their business, and that learning process really never ends. And I think it's a key part of success. This is probably pretty obvious, but in all of this, intellectual property is critical. You have to have both an understanding of that world as well as you have to have some. Otherwise, it's very easy for people to disregard you, and it takes away leverage. And then lastly, and this is really my last slide and my last quote, it's in all of this, style and professionalism do matter. Often as scientists, we think that it's all about the substance, and I think without it, you can't do anything. But for example, one of the metrics that I've learned from actually smarter people that I've worked with is that at each engagement, when you start, for example, setting up a company, you want to come across as if you're already at the next step. And you want to have your slides and your standing as high level as you can possibly make it, because these impressions do last, and therefore it's really the stylistic element that is also a key component of success. But again, not to be taken in isolation. Without the substance, you can't do it. So with that, I want to close off and look forward to if there's any questions, or and certainly look forward to the panel later as well.
Thank you so much, Dr. Vanderburg, for your informative presentation. I think we all learned a lot about not only the academics, but also the business aspect of gene therapy. We have a couple of minutes till 2:40 EDT to take some questions. So I'll open it up to the audience. You can type your questions within the chat or the Q&A section, or even feel free to raise your hand using the option in your toolbar below if you would like to directly address Dr. Vandenberg, and we will enable you to unmute yourself. So I'll just give the audience a couple of minutes to come up with any questions, and if not, I definitely have questions of my own. Okay, so while we're waiting, I think I'll ask one or two questions because we do only have 2 minutes. My first question is, you predominantly mentioned viral vectors. My question is, are any of your startups interested in non-viral vectors? And what is your reasoning for predominantly sticking with viral vectors over non-viral vectors for gene therapy?
Yeah. Our general thinking is that we're at some level technology agnostic, and that may seem strange because I think almost everything I mentioned has related to actually one particular viral vector technology, AAV. But if something shows up that performs better for the goals that we are pursuing from a disease perspective, we will use it. But AAV, for particularly in vivo gene therapy, for a number of targets, the ear, the eye, some of the drugs that I mentioned that have found approval, from at least my perspective to date, even though non-viral approaches are incredibly promising, and certainly we can see that right now with mRNA, for example, for the types of diseases we are pursuing, which require durable intervention, AAV is kind of the best workhorse till now. It has limitations, right? And in part, we try to, within our box, overcome these limitations, but we are fully aware that we won't be able to overcome all of them and that there are likely going to be, hopefully, emerging better technologies. And then we will probably move into those technologies. But for in vivo gene therapy approaches, certainly outside of the liver is obviously something that many technologies can go into. We stick with AAV because it works and it's safe.
Yeah, I think that's an excellent way of looking at it, not being technology agnostic, because it's easy to get stuck in using that one technology once you're used to using it and have that expertise.
Absolutely.
So thank you so much. And we're super excited that you will be continuing with us at the panel discussion later on. And we will definitely ask you some more questions then. But for now, I think we'll redesignate you back to attendee status so we can continue with the next presentation, and looking forward to speaking to you soon.
Great, thanks so much.
Thank you. So our next presenter is Dr. Christina Mcburney. She's currently not present, but so I'll give her a minute or two. If not, I do see Dr. Jason Acker here, who is also our third presenter. So Dr. Acker, please let us know if you would be okay to shift your presentation to an earlier time if Dr. Christina Mcburney is not able to make it on time. That's excellent. So just keeping in mind of time, I think we will go ahead with Dr. Jason Acker's presentation. So we will now promote Dr. Jason Acker to speaker status. So you can present your video and audio. And I will give a brief introduction to Dr. Jason Acker. Dr. Acker is a senior research scientist with the Canadian Blood Services Centre for Innovation and a professor in the Department of Laboratory Medicine and Pathobiology at the University of Alberta. His research focuses on understanding the response of cells and tissues to ex vivo storage and the development of methods for their preservation and use as therapeutic products. Dr. Acker is the author of more than 200 publications, 10 book chapters, and holds 12 patents in the area of cell preservation and microfabrication. He is actively involved in consulting with and advising companies and organizations developing biobanking and cell therapy programs, and is also the co-founder of 2 startup companies. We're super excited to have you, Dr. Acker, especially in the Law and Business Hall. I think you're an excellent fit. So the floor is yours, and looking forward.
Excellent. Thank you. And thank you for the invitation to present today. It's a really exciting opportunity to speak about some of my experiences looking at transitioning technology from bench to business. And I just want to thank Dr. Vandenberg for really providing a lot of case study in his experiences, because they do, again, reflect a lot of the challenges that we have sometimes in taking technology out of an academic research program and moving it into a business. But again, a lot of the messages from his talk will resonate well with what I share with you today. All right, so what I'd like to do just briefly over the next 30 minutes is describe some of the opportunities that exist for technology commercialization in academic research. And I'll use my experiences with two new codes that we brought out of some technology we developed, just as case studies to share with you my experiences. And then hopefully you'll be able to reflect on some of the barriers that exist with science entrepreneurship in your organization, whether you're at a university or whether you're at in government or in an organization where commercialization and entrepreneurship is encouraged. And I'll use 4 Ps as a way to introduce you with some of the concepts that you might want to consider as you're embarking on this journey. So just a little bit more on who I am. You know, I have a very traditional academic background. I did all 3 of my degrees, science degrees at the University of Alberta in Edmonton. I then had the opportunity to go to Boston and work at the Center for Engineering and Medicine at MIT and Harvard for a number of years. And then got on a very traditional academic treadmill, you know, where I started as an assistant professor in lab medicine and pathology at the U of A. I was fortunate to have a primary appointment with Canadian Blood Services, where I still work as a scientist. It's a very unique relationship between industry and academia, and I'll talk a little bit about that as I go through and share with you my experience. I followed sort of the academic treadmill, you know, doing research, serving on committees, supervising students, teaching. But in 2004, I decided that it was time for me to really do something different. And that something different was really pursuing a business degree at the University of Alberta, and which really embarked me on a path of technology entrepreneurship and commercialization. So just a little bit about my why, you know, and this will give you some context for you know, what motivates me and how I approach my academic research program. You know, I really do follow a very standard innovation pathway where we have ideas that we do research, and I do the research both within my university lab and with my position at Canadian Blood Services. And that research results in new knowledge, and we publish and we present and we do all the standard academic things around sharing that new knowledge. And in a lot of cases, you know, that's the extent of an academic career, is that you really do stay in that discovery phase. Although more and more, I think we're being asked, and I think we're being encouraged and motivated and incentivized to actually think about the next steps. And for me, for a lot of the work that I do in clinical medicine and laboratory pathology, a lot of that leads to changes in practice. And that changes in practice can be done through a variety of outlets and organizations that I'm involved in. Some of them are with Canadian Blood Services. Others are with, you know, groups like Alberta Health Services, or in the case of transfusion medicine, some work I do at the international level looking at best practices around transfusion medicine. But there's equally the opportunity to take that knowledge and through an organization that we have at Canadian Blood Services called NetCAD, or the Center for Blood Research. We have the opportunity to actually develop those ideas into actual products or services that have an impact in transfusion science or in cell therapy and other applications. So that's the path that's also been very exciting for me from a career perspective, where I've actually taken knowledge and put it into a variety of new codes. And I'll use Panthera Cryo Solutions as a case study today, because it aligns well with the stem cell theme for this conference. But equally, I could talk about my expertise with Aquila Diagnostics, which is more of a traditional diagnostics company. So the first P that I want to introduce everyone to is the idea of a product, or in some cases a service, but in a lot of times we're thinking about that product that we're actually going to be producing. And the first lesson I want to sort of instill is that, you know, even though you have a great idea, you know, you've done some phenomenal, groundbreaking discovery research, that doesn't always equate to a commercializable product. And I'll talk about what that means in a minute. And I've been involved in a number of groups that have evaluated technology, and I work with a lot of mentorship programs where I meet with graduate students and postdocs and in fact, early stage career faculty members. And no matter how excited the research is about the product or about the discovery that they've done and how novel and innovative they think it is, or how high impact the journal is that work is published in, that doesn't really mean that there's a commercialization pathway for that idea. So you need to really think about what that product pathway is. So I'll talk a little bit about the Panthera technology. So this is a technology that was developed by Dr. Robert Ben at the University of Ottawa and his team. And it's based on using very conventional carbohydrate chemistry and rational design and high throughput screening to identify compounds that interact with and control the growth of ice. And from a cell therapy perspective, when we think about bringing cell therapies to market, cryopreservation invariably comes into the discussion. And I'm one of the few, I guess, experts in Canada that have graduate programs and research facilities and labs dedicated to understanding the cryopreservation of cell therapies. And so working with Rob, he came to us with this really innovative approach. This was small molecule carbohydrate-based chemistry that had very potent ice recrystallization activity. And ice recrystallization is that process where you have small ice crystals like seen in the bottom square that grow into these very large ice crystals at low sub-zero temperatures. And it's this recrystallization that occurs either during the freezing process or during the thawing process that can be extremely damaging to cell therapies. Most common cryoprotectants that we use in the industry, like dimethyl sulfoxide or glycerol in the case of red cells, have very little ice recrystallization inhibition activity. So they aren't able to prevent this from occurring. So this was potentially a technology that we could see some commercialization path right off the bat. But the first question we have to ask ourselves, and the first question you should probably ask yourself is this a novel idea? Yes, publications are one phase of that assessment, and that's the peer review. That really is more about the science. That doesn't mean that it's a patentable invention. So the question you have to ask if you want to pursue IP, and that is one path, and IP in a lot of cases is a minimum criteria to pursue a technology commercialization, but it's not absolutely necessary. But you want to address this right off the bat. Is this a novel idea patentable? And the best way to do that, obviously, is to go through that patent process. And that patent process can be very lengthy and time-consuming and challenging, but it's one where, again, it really helps you determine whether you have a novel idea. And the definition of patentability comes into play here, that you obviously have to make sure that your idea adheres to. But once you've demonstrated that it's novel, how do you get that product? How do you get from the idea to the product? And this is where really, as scientists and as academic leaders, our creativity doesn't have to stop with the invention. Here's where we can actually be very creative in thinking about where this technology may actually have application. So in the case of our ice recrystallization inhibitor technology, we were publishing this after we had patented it, and we're starting to generate a lot of publications. And we're really looking at initially cell therapy. So this is work that Rob and his team and my graduate students did a few years back, where we're actually building up the technology to actually be useful for the cryopreservation of specific cell therapies. Rob and I started working together in late 2014. So it took us about 2.5 years to take that basic discovery and bring it into something that was actually what we would consider a product. And that was after, as you noticed on the sign, after Rob and his team coming to my lab and us working collaboratively for at least 14 trips over a very short period of time. So a lot of collaborative work that can go in when you're actually pursuing this idea of a product for that technology. Now, our productization of this ice recrystallization technology really is around building it into something that an end user would use. So, how would an end user use a chemical compound? Well, you have to think about things in the case of cryoprotectants like formulation, packaging, the buffer system, and other cryoprotectants that you may want to add to it. And then as you're doing that, you really have to think about what kind of data sets, beyond just efficacy data sets, that you would need to have to convince an end user that you have an actual product that is useful. So is your chemical synthesis process scalable? Are the chemicals stable? What quality control do you put when you're manufacturing this? What's the toxicity? What's the purity of these compounds that you're actually producing? And then in the case of our application, we're really looking at cell type specific cryopreservation protocols. So what kinds of cells can you use this technology with, and what kind of value proposition does it provide? So in that case, selection of the IRIs. The IRI technology we developed is not just a single compound, it's a family of compounds. So which one works best with which cell? What's the toxicity of those ice recrystallization inhibitors? And again, Is it potent? Is there some sort of process optimization that you have to go through? And ultimately, you have to get it into a form factor that someone can actually use and evaluate. So that's a path that we had to take with developing this technology. And some of the things that are obviously really important to consider: talk to people. The best way to learn about where some opportunities might be for your technology is really to get out and talk to the people that are going to potentially use the product. Getting that voice of the customer early is really critical to help define that product development path. Often, working in academic research labs, we don't necessarily have those connections, which make it can sometimes very difficult, A, to identify where this technology might have value, but also B, to make some really poor assumptions about what those customers might ultimately want in a product. And as a result, we go down a path that really isn't helping move this closer to commercialization. Other things to consider, just as an academic researcher, it's harder to get product development funding with traditional discovery-based grants. So you really have to be creative in seeking out alternative sources of funding. And these do exist. They exist through industry, they exist in government, they exist in training programs and staff programs that you can bring in to help support this work. More and more, the traditional tri-councils here in Canada are looking for opportunities to take technology out of the academic lab and move it into commercialization streams. So there is support out there. So be creative and seek out those funding streams. But remember, those grant programs are not looking for discovery. They're really looking for closing the gaps on the knowledge that you need to have in order to bring this product to market. The other thing I just want to, again, caution you early on is that, publishing, which is obviously so critical for a lot of graduate students and trainees, in order to actually meet the program requirements. It's important for academics to demonstrate productivity to faculty evaluation committees. But publishing the results early can jeopardize your ability to get the intellectual property. And publishing product development results are sometimes very difficult because the discovery-based journals don't want to see that data. So you can either publish and jeopardize your business opportunity, or you're not able to publish. But in the end, your traditional academic output of publication count may decrease during this period if this is something that you're actually pursuing. So you need to reflect on that and make sure that you're comfortable with that, and have some very important conversations with your supervisors, in the case of academic, your department chairs, and hopefully the evaluation programs that you're going to be going through will understand that this is just a reality of the path that you're on, and that that the impact that it will come from this work will not be seen in publications, but will be seen in other means and other forms. So the next P that I want to talk about is policies. So the lesson number 2 is really around knowing your rights before you start, and knowing what the rules are early on. And this is something that's so important. I think Dr. Vandenberg mentioned it as well around intellectual property, but it's more than that. One of the things that I learned again early in this process and through my pursuit of a business degree, business 101, everything is negotiable. So take that into your conversations with people that you need their support in order to actually achieve what you're trying to achieve. So always remember, everything's negotiable. Here in Alberta, there's a number of programs that help support early stage entrepreneurs with either coaching and mentoring. So organizations like Startup Edmonton or eHub or Chinook, Entrepreneur Challenge in Calgary, or the Hunter Center. Each of our institutions in Alberta, University of Alberta, University of Calgary, have very detailed policies around intellectual property, policies around what you can do on and off campus in terms of housing startups. Get informed with what your local institutional policies are. It's really critical to have that knowledge and not go down a path that could get you into trouble early on in the process, or worse, cause you to have to abandon your efforts because you've run aground of some policy or procedure. As graduate students, many universities have different policies around intellectual property ownership, so make sure you're knowledgeable about what that is before you actually go down this path, because it can actually bite you later on if you don't understand what your rights are and what your responsibilities are for reporting to the institution. In Alberta, we are, as a province, undergoing a provincial review of our intellectual property policies. So stay tuned to see what that does. Hopefully, my hope is as an entrepreneur that the government comes out with policies that are, again, encourage intellectual property being leveraged from the academic institutions and don't put any more barriers into what the policies currently are. But I know in Ontario, again, different jurisdiction in terms of policies. So check out what your local policies are. The next P is really the plan. A good idea, is not a commercializable product, and it's also not a business opportunity. So just because you have a good idea, and yeah, there might be a product that you can actually produce, how are you actually going to translate that into a positive value? And that positive value could be sales, or it could be, in the case of a clinical product or procedure, it could be the impact on the patients and patient outcome. How is that actually going to be achieved? And again, no matter how good your product works or how good your story is, really, in order to be successful at this endeavor, you really have to make a business out of it. And a business is not the product, and a business is not the idea. A business is much more than that. From our perspective, from a Panthera perspective, we started with developing our technology to aid in discovery research, the cryopreservation and biobanking of cells that are used in diagnostics, and as reagent cells for production of other clinical cell therapies. So we've also then moved that into clinical cell therapy programs where we've used our technology for the cryopreservation of immune cells, from T cells to CAR Ts, T cells, Tregs, as well as MSCs, iPSs, and a variety of other cells. And then to do that, we've obviously had to work with partners. So we're working with a number of organizations around the world to bring this technology to market. But you also need, a pathway. Where's this going to go after that? And we've taken this technology now into engineered tissues, and we've even taken it into whole organ cryopreservation with a variety of groups in Canada and in the United States. So we have a pathway for this technology that, again, builds value to the business that we're actually creating behind it. Sort of a subcomponent of this is really, I like, this quote: "You can't observe a lot. You can observe a lot by watching. That said, you couldn't know what you didn't know, but now you know," which is really a very complicated way of saying you've got to get out there and talk to your potential customers. You really have to understand what the problems are that you're trying to solve, and you really have to understand how to best position your technology, what data they're going to want to see, what form factor they're going to want to see it at, and what price point they're willing to pay for it. Because often, you can have a fantastic technology, but the price point is that the customer will accept is just not there, and as a result, you can't build a business off of that. So you have to, maybe abandon that and look at other opportunities. From our perspective, again, getting out and talking to the customers has been really important. Dr. Eric Woods at Ossium, states it well that the value proposition for our technology is such that the current paradigms for cell cryopreservation just aren't good enough, and that we fundamentally need a new technology like the one that we're bringing to market. And that, again, gives us some credence to the direction that we're taking from a business perspective. And then we can work with other partners like Stem Cell Technologies in Vancouver, where, again, we've been working with them to evaluate our technology. And they, again, understand that there's a strong demand in the market for improved cryopreservation of cells, and that they see our technology, again, as a major improvement to the technology. So when you can start to have these kinds of conversations with thought leaders in the area, it really, again, helps build support for what you're trying to do and what you're trying to achieve. Lesson number 4 from, again, the planning phase is you need a business plan to focus your thinking and defining your actions, and your investors want to see this. And this is often, one of those areas where, as academics, we start to, back away from our interest in being involved because of our lack of familiarity with working in this space. And I want to, maybe reassure everyone that, a business plan is not that different than a grant proposal. A business plan is really a pitch, a document that helps outline, what the opportunity is, how you're going to pursue that opportunity, what resources you need to bring to that opportunity, and what you hope to see as an outcome. And that's, so writing a business plan is not that different than writing a grant proposal for many of us in the academic world that do that on a regular basis. So don't be afraid of this. Just recognize that there's going to be different language that's used, but the process itself is actually something you're very familiar with. And ultimately, you need this business plan because at some point you're going to be going and asking investors or individuals to help continue to fund this action that you're taking. And as Dr. Vandenberg said, around professionalism and style, you really only get one shot to convince dragons that you know what you're talking about. And you can't make data up. You can't not know. You cannot have incorrect answers to some of the questions. And really, you have to have your A game in place when you're actually going in front of investors and trying to convince them that your opportunity is one that they should take a look at and they should invest in. Because individuals know that are investing, that they're seeing hundreds of opportunities at a time, and they have the pick of which ones they want to pursue. So you really have to help have yours stand out. Much like writing a fellowship application or a studentship application or a grant. You need to really make your idea and your concept stand out, and you have to have the data and you have to have the evidence to support everything that you're actually saying. And the final P is really, I guess it's a two-partner P. It's the people and the partnerships that are going to make all of this happen. And so lesson number 5 is really, if you're embarking on this as an early part of your career, the learning curve is incredibly steep. So to be successful, you really need to find partners who have reached the summit before. So, you would not embark upon climbing Everest without, leveraging a Sherpa who's done it, tens or hundreds of times before. That really reduces the risk that you aren't actually going to make it to the summit. You actually have someone there who can lead you, guide you, and help you avoid all the pitfalls that are going to happen along the path. In the case of science, just as science is a team effort, these are some shots from my research team and then Rob Ben's research team. So is building a company, and building a company does require partnerships and does require you to reach out and bring in support and help. I love this quote because Jim Rohn says, "You are the average of the 5 people you spend the most time with." So if you are not spending time with other entrepreneurs and individuals interested in commercialization, how do you expect to be successful in that space? So find opportunities. And here again in Alberta, we have some, Startup Edmonton, eHub, the CDL. We have venture mentoring services, Mentor Alberta, Tech Edmonton. There's many of these kinds of mentorship organizations that you can participate in, and just listen, network with, and pick up connections that are really going to be valuable as you start to go down this path. And I can't underestimate the value of mentors. And this is something that I tell all of my graduate students and postdocs early in their careers. But it's something that I also tell tech entrepreneurs who approach me is you really need to seek out a mentor who has relevant experience and whom you can develop a relationship with. As a student, as a trainee, you really have a unique opportunity. And that's because most individuals that you might want to mentor with, whether it's a successful entrepreneur, whether it's an academic who's done this before, whether it's someone in industry, whether it's someone in government, approaching them as a trainee and really gives you an advantage. When students approach me and ask for my help, I rarely say no. But if someone from industry approached me or someone from government approached me and wanted my help, I'm less willing to support them, only because I was a student before and I want to give back to the training of the next generation. So leverage that status and use it effectively. There was 3 women in my MBA class who were wizards at this. Early on in the program, they got together and they created a pact that every month that they would go out and arrange a coffee date with someone, a high-profile individual in business or government, who they wanted to talk to. And they leveraged their student status. They would get a hold of the executive assistant for that person and introduce themselves as students who wanted to interview and talk to that individual, and would they be willing to meet for coffee. And they got access to individuals that many of us would be astounded to, that they actually got access to, and were able to get an hour of their time to really learn from those individuals. And that is experience that you can never pay for. So again, take advantage of it. But also when you're approaching, or when you're starting to identify mentors, understand what you can bring to the relationship as well. Because, you know, it's really, that'll help, again, break down some barriers and help, you know, again, add some value to the relationship. So, you know, consider that. And if there's questions about this, I'm happy to answer them at the end. So just some final messages. You know, early assessment of an idea and technology and the market need is critical to building a story that will be compelling to investors. You have to do it early. And I love Dr. Vandenberg's adage that you want to fail fast. And that is true. You want to learn quickly whether your idea, your discovery is potentially commercializable, and if it is commercializable, is there a business model that you can pursue with this? If not, you want to learn that early. You don't want to invest time and energy and years of your time, of your life pursuing something that will never ultimately go somewhere. To do that, though, you really have to do your homework, and you have to verify every assumption that you're making about the market, about the technology, who's going to use it, the business opportunity. And that is a skill that, again, as you get more experience with this, it becomes easier, but it's one where a mentor or a group of mentors can really help you by asking questions and forcing you to find out what the answers are. And I'd just like to, again, encourage all of you. Now, there really is a need for Canada for individuals with experience in life sciences, from moving technology out of your research labs. So be bold, look for those opportunities and pursue them, because there's a world of opportunity out there. And I've been very fortunate to have a career where I've been able to leverage both my academic research program, the development work I work with Canadian Blood Services, and the company work that I do in order to see value. So with that, I'd be happy to answer any questions.
Thank you so much, Dr. Acker. That was an excellent presentation. I think it was a little different from what we generally expect, and it provided us some really useful advice as we kind of navigate our own career journeys. So now the floor is open to the audience in case they have any questions. Feel free to use the chat or the Q&A feature, or even raise your hand in case you wish to address Dr. Acker directly. We have time till 3:15 to do so, which is when Dr. Christina Mcburney will begin her presentation. So I'll give the audience a minute maybe to ask you any questions. And if not, as with the previous presentation, I definitely have a few that I'd be more than willing to ask. Okay, so I'll give them more time, but I think till then I'll ask my question. So Dr. Acker, in undergrad and high school, we're typically taught how to use techniques, and we're not really thought to think about the storage techniques, the tools, how to improve them, how to think about those. So what kind of encouraged you to think beyond therapy or the typical applications of scientific research?
Yeah, it's a good question again, and the best way to do that is get involved in organizations. So one of the, again, privileges that I have is working with Canadian Blood Services. We collect, manufacture, store, distribute blood products for Canadians. And you would think it'd be a relatively straightforward process, but there's a lot of things that we do, and there's a lot of areas of transfusion medicine where there really is a gap in our evidence to support practice. So working with frontline physicians who are transfusing, the transfusion doctors, the blood bank techs, our own manufacturing and quality control people, it's very easy to identify opportunities where there's a gap in knowledge. So a lot of the ideas that have come up particularly on the diagnostic side and in some of the blood product quality manufacturing work that I've done over the years really comes from people just saying, no, we're having a problem. This isn't working. No, how do you fix this problem? And that's what I love. Again, I love, I've really focused my career around being able to apply science and technical skills to solving problems. And if someone asked me what my job description was, what is the job description of a scientist? Well, we really are professional problem solvers. That's what we do. So, use that process of solving problems, whether it's a gap in knowledge or a real technical problem, to see the opportunity for a new device, a new technology, a new service model. And that's where, again, when you can think not just how to fix this, but if I fix this, what value would it have? And as soon as you can start thinking about that, then you're starting to think like an entrepreneur. How do I actually create value from this? And it used to be that, there was a lot of, there's a gap between academics and industry, right? And you had to make a choice. I'm going to do a PhD in academia, or I'm going to do a PhD in industry. Well, I think the reality is you don't have to do one or the other. You can do both. And if you do both, you're going to be a lot more successful, because you're going to be able to leverage, again, the opportunities, skills, resources across the continuum. So, take advantage of that. I think there's really an opportunity to do that. It's less looked down upon. Again, as an entrepreneur, we talk about making money. No, that's not a bad thing. Making money is part of creating the money is then the fuel that you use to create new ideas and new technology that solve more problems. So money is just the fuel that drives that engine.
Yeah, I completely agree. That makes a lot of sense. And it's a fresh perspective. So thank you. And my other and last question, just looking at the time, is you kind of mentioned an entrepreneurial mindset. What intrigues me is this something that people just have, or is this something that you can develop over time? And if you can develop it over time, what are some key resources or key conversations that one should have to develop it?
Yeah, great question. It's like the argument, are you a leader, or are you born a leader, or can you develop to be a leader? Are you born an entrepreneur, or can you develop to be an entrepreneur? And I'm very much, environment versus nurture versus nature. I'm very much, it's a little bit of both, but I think, finding mentors, finding people that can challenge you to think differently is really a key to that. So, I was very fortunate during my postdoc to have a supervisor who was, a brilliant scientist and very accomplished, on the academic side, but also, was in entrepreneur. He was an entrepreneur. He had many companies, and he was able to translate those ideas from knowledge to practice. And I learned a lot from him, and really then, he seeded in me the thought that, if he could do that, why can't I? So, finding those mentors is going to be really helpful. But also, when you're, even as an undergrad or as a graduate student, look at the problem you're trying to face right now, whether it's, a specific research project, and ask questions like, who's going to benefit from this? And then how would they benefit from this? So, if I solve this problem, okay, I'm going to help, if it's a cell therapy, I'm going to make a cell therapy better that's going to be able to treat, a disease. Okay, well, that's good. The technology's there, but how? How is that going to happen? Because there's no good technology, there's no medical device, there's no good discovery that, if it is not commercialized, will see value. There's technology that, we see it all the time. People come up with great ideas that just sit in papers that never actually get translated. So, when you're thinking about, if you're working on something, and you're really excited about science, just pause and say, okay, what would be the next step? I'm going to share this. I want to get the paper. I want to get my degree, but then who's going to use it and how? How would they do that? And if you can start to explore that how, you're really starting to think like an entrepreneur, because I think that's really the next step is we're really creative on the discovery side. I think we need to involve that same creativity to try to address that question of how.
Thank you so much. And that you brought up mentorship, and I'm really glad that you kind of chose to take part in our conference and provide some limited mentorship through your talk. So thank you once again. And with that, I would like to wrap up the second session in the Law and Business Hall. And thank you once again, Dr. Acker, and we will now redesignate you to attendee status. And please enjoy the rest of the conference. I would now like to welcome Dr. Christina Mcburney, who will be our third speaker today. I would also like to promote her to speaker, so please be prepared to share your audio and video. Just to quickly go through the format of this session, after Dr. Mcburney's formal presentation, there will be a 5-minute Q&A that I will moderate. Attendees, feel free to type your questions in the chat or the Q&A feature, or you may even ask Dr. Mcburney your questions directly by raising your hand at the end of the session, and we will allow you to unmute yourself. So now that the logistics are out of the way, I would like to introduce our speaker, Dr. Christina Mcburney. Dr. Mcburney is the Manager of Scientific Communications at the biotechnology company that Dr. Acker just mentioned as well, Stem Cell Technologies. Dr. Mcburney's work has primarily focused on creating quality educational and news content for those in life sciences fields. Her extensive experience in research communication and education is highlighted by her work in stem cell programs such as the Stem Cell Podcast, Science in the City, and Science News. Please join me in welcoming Dr. Mcburney.
Thank you so much. And I'm sorry that I'm a little late. The great thing about virtual conferences is you can get people presenting from all over the world. Exactly. The downside is that sometimes time zones get a little messed up, but I'm here now and I'm just gonna share my screen.
Yeah, for sure.
All right. So again, thank you everyone for having me. I'm really excited you're all here. And today I'm going to be talking a little bit about science communication and social media. So I thought I would start by introducing myself. So my name is Christina Mcburney, and I am the Manager of Scientific Communications at Stem Cell Technologies. And what I really do is manage some of our, what I call non-traditional scientific communications services. So as mentioned, this includes the Science in the City program, which is a series of life science news websites found in Vancouver, Seattle, and Boston, and hopefully Toronto soon. The Stem Cell Podcast, which is a bi-weekly podcast discussing the latest stem cell research, and the Science News program, which consists of 21 newsletters in various areas of stem cell and immunology research, just to keep you up to date with what's going on in those fields. Now, altogether, my team at Stem Cell manages 35 social media channels that have over 200,000 followers. And so suffice to say at Stem Cell, we do know something about social media and science communications, which is of course why I'm here to talk to you all today. So I thought we'd start by talking about some traditional channels of science communication that scientists use to share their research. So as an example, one of the main goals as a scientist is of course to publish your findings in a scientific journal for other researchers to view. As well, you might attend conferences such as this one or department events where you present your research in the form of a poster or a talk. Now, all of these channels have many advantages. They're fantastic, but they do all share one limitation, which is that they cater specifically to scientists. So namely, posters and talks, of course, are accessible only to the researchers who are fortunate enough to be able to attend those events. And scientific journals, although to an extent more accessible, are of course full of technical jargon and details that the regular layperson just isn't going to be able to decipher. So that creates a problem because, as we're going to discuss in a moment, the public needs to be able to understand science in order to inform decision making. So for this reason, I like to say that science isn't truly finished until it's communicated to the public, because without this last bit, it can lead to situations like this one outlined here, where scientists say apples and the public says oranges. Now, some of you might think that this apples and oranges comic is a bit of an exaggeration, although I think we definitely see it a lot right now during the pandemic. But I did want to include an example from a recent study that did garner a lot of media attention, in part because of some miscommunication. So some of you might remember the NASA twins study that was published in Science back in April of 2018. So in this study, it involved comparing both physiological and molecular changes that were experienced by two identical twin astronauts, so Scott and Mark Kelly, over the span of a year in which Scott was on a mission in space and Mark was on Earth. And of course, the hope here is to identify molecular or physiological traits that could be affected by time and space. Now, the researchers conducting this study did discover a number of interesting changes in Scott that were attributed to his time and space. And I've included an excerpt here from the paper that discusses one of them. So as you can see, this says, a distinct subset of genes did not return to pre-flight levels within 6 months of return to Earth including genes related to immune function and DNA repair, representing candidate genes that may be altered for an extended period of time as a result of long-term spaceflight exposure. Now, as scientists, it's obvious that the authors, when they talk about genes that were altered, are referring to transcriptional alterations, so the expression levels of these genes. However, as this paper was not released with a clear explanation of the results for the media, they thought that this meant that 811 of Kelly's genes had been physically altered, or in other words, mutated by his time and space, which is obviously a very, very different thing. And this was reported really wildly by the media. So I've included a couple of examples of headlines here. You can see that they wrote, astronaut Scott Kelly now has different DNA to his identical twin brother after spending just a year in space. Astronaut Scott Kelly's DNA no longer matches identical twins after a year in space. NASA astronaut twins Scott and Mark Kelly no longer genetically identical after space trip. So NASA and the researchers who conducted the study wound up having to obviously put out a mass correction explaining that Scott's DNA had not in fact been mutated. It was still genetically identical to his brother Mark's. But I do think that this example does a really good job of highlighting the importance of communicating science to the public and how easily results can be misinterpreted if that communication isn't effective. So now that we hopefully all agree on the importance of communicating research to the public, the obvious question is, well, how do we do this? And I'm going to try to convince you today that social media is at least one of the platforms that we should be turning to. So I'm sure you're all familiar with social media, which is of course defined as websites or applications that enable users to create and share content or participate in social networking. And in Western culture, we have what we call the big 5 social media networks. So these include Facebook, LinkedIn, Twitter, YouTube, and Instagram. And each of these platforms allows users to share content, including science, through text, photos, videos, or other types of media. And really, one of the greatest advantages of social media as a platform for communicating science is that it's one of the most accessible platforms out there. So here's a bar chart that's showing the number of active social media users worldwide from 2010 to 2017, and then projected to 2021, which is of course this year. And what you can see is that by 2021, social media users worldwide are expected to reach 3.02 billion. Now, for context, that is 10 times the US population, and it's nearly half the global population. And this is just active social media users. So this means that the number doesn't include those users who have profiles, but maybe haven't logged in for a while. So you can really see how social media truly is a highly accessible channel of communication that we might want to take advantage of. And as mentioned, there are a number of different social media networks available that researchers can use to share the research. But today, we're really going to be focusing in on Twitter. And the reason for this is that Twitter ranks the highest amongst researchers for sharing scientific content. So what you're looking at here are the results of a survey conducted by Nature News, in which they asked a group of academic scientists which social media networks they preferred using to either A, share links to authored content, B, post work content, C, actively discuss research, or D, comment on research. And as you can see, in all 4 cases, they found that Twitter was the preferred platform, beating out not only Facebook and LinkedIn, but kind of in a funny way, also ResearchGate, which is actually a social media platform that was created specifically for scientists to share their research. So considering this, it's not really surprising that studies also show that scientific research is increasingly reported on Twitter over other platforms. So this is taken from a study conducted at the University of British Columbia. And what the researchers found was that back in 2005, the most popular platform for reporting scientific research was blogs, followed by news outlets. However, by 2015, you can see that Twitter had started to grow quite a lot. And you can imagine by 2021, this has increased even more. So what is Twitter? Well, many of you are probably familiar with it. It is a social networking site, and it allows users to post short 280 character messages that are called tweets. So you can see an example of one on the left-hand side here. So there's three main things that make Twitter so great for sharing science. So the first is that those tweets are short. Again, they're limited to 280 characters, which really forces you to think about the most important point that you're trying to get across. So you're not going to be able to get super convoluted with an entire paragraph. It also makes it really great for consuming science updates, as the tweets are really easily digestible. Second, Twitter gives you the ability to post multimedia content. So you could link to a scientific article, you could post a figure, you could include a video of cells growing in the lab. You can even post a poll if you're trying to troubleshoot an experiment. And third, unless you have a restricted account, which most people don't on Twitter, anyone can view and interact with your tweets. So that means that you can have researchers in, say, Toronto, reaching people all over the world. So it's really, really accessible. And the other great thing about Twitter is that it takes very little time out of your day. So this is data from the Bureau of Labor Statistics, and they found that the average time spent on Twitter during a lifetime is just 18 days. This is compared to other social media platforms like Instagram, where it's 8 months, Snapchat 1 year and 2 months, Facebook 1 year and 7 months, or depressingly, YouTube 1 year and 10 months. And the reason for this isn't that people aren't using Twitter. It's just that it takes so little time a day to use it because of the short nature of the tweets. So Twitter still has 330 million monthly active users, 100 million daily active users, 140 million tweets published per day, and almost 500,000 new accounts created daily. It's really just because it takes so little time to publish and also to digest other tweets. But this raises the question, who is viewing these tweets? You know, are they other scientists? Are they members of the general public? Because depending on who you want to get your research in front of, this might matter. Now, unfortunately, Twitter doesn't compile this type of data on followers, whether they're scientists or not. But fortunately, this is something that researchers have looked into. So this is data taken from research published at Simon Fraser University in BC. And they looked at the Twitter accounts of a group of 110 academic scientists and determined what percentage of their followers, and therefore the people viewing their tweets, were other scientists, the general public, or other groups. And interestingly, what they found is that for academic scientists with under 500 Twitter followers, over half of those followers, or about 65%, are in fact other scientists. Now, as their followers grew, as you can see here, this number was reduced, bottoming out at 46%, which is still about a 50-50 split. So this really makes Twitter one of the only social media networks that can target both scientists and the general public to an equal degree. So, given everything that we know about Twitter, how can you maximize your impact while communicating science on the platform? So the very first rule of really any form of science communication is to know your audience, because your audience is going to dictate your messaging and your word choice. So what do I mean by that? Well, let's imagine 4 different audiences that you may want to target depending on what science you're trying to communicate. So scientists, patients, policymakers, and the media. Now, these groups are hopefully all going to be interested in your research, but there's going to be different aspects that interest each. So maybe to help better demonstrate this, let's go back in time and imagine that we're researchers working on publishing the first ever study showing genome editing in human cells using CRISPR-Cas9. So if we were presenting this research at a scientific meeting to researchers, you can imagine that the 2 questions that other researchers would be the most interested in having answered would be, does this CRISPR technology work? And how is it going to affect my own research? Is it something that's going to help advance it? Patients, on the other hand, will be primarily interested in how this research could affect their health. So could CRISPR-Cas9 offer a cure to disease? And if so, are there any side effects? Policymakers are going to be interested in how CRISPR-Cas9 will affect the public and the sentiment from the public. So they're going to be asking, are their constituents fearful of gene editing? Are they excited about its potential? And then, of course, there's the media. Now, when it comes to the media, they're always just looking for a good story. So something that's going to attract an audience and increase their viewership. So basically, they're going to be asking, is CRISPR-Cas9 sexy? And so you can imagine that as a science communicator, when you're writing a tweet, you need to consider who it is that you're trying to target with that tweet and what it is that they really want to know about in order to engage them. Similarly, when communicating to the public, it's important to make sure that you avoid jargon. So for any of you that don't know, jargon is defined as special words that are used by a particular group and are difficult for others to understand. And as scientists, we use a lot of jargon. So this, of course, includes scientific words, so things like heterochromatin, progenitor cells, immunoprecipitation. But it also includes a lot of regular words that have a different meaning in science. And I've included a few of these in this table here. So as an example, in science, when we hear the word culture, we know that that refers to the process of growing cells. But to a layperson, they're only going to know the definition related to social behavior and norms in societies. Another really good example is clone, because the public kind of has a scientific understanding, but not really. We know that it means the replication of recombinant DNA, but to the public, they just kind of think of like Dolly the sheep. So it's not that we want to dumb it down, but we do want to present it in language that anyone can understand. And so a research group at the Karolinska Institute actually performed a study a few years ago looking at the use of scientific jargon by researchers in academic journals and how it's changed over the years. And really interestingly, as you can see here, what they found is that the readability of abstracts has actually decreased substantially over the years, and that this is due to a decrease in what they call common words that are used, and an increase in what they refer to as science-specific common words. So these are common words that are scientific, that people understand, things like DNA or cell, but also general science jargon. So these are going to be the words that the general public just doesn't understand. And from these results, the authors of the paper concluded that more than a fifth of scientific abstracts now have a readability beyond graduate level English. Now, when it comes to the abstract of a paper that is meant to be read by other scientists, this might or might not be an issue. But considering that only a small proportion of the population has an education beyond graduate level English, we as scientists need to be careful not to use this type of jargon when communicating to the public. So once you've determined who your audience is and you've ensured that you're not using jargon, you also need to think about how you want to frame your message. So what do I mean by that? Well, let's use the example of a doctor discussing a potential surgical procedure with a patient. So when discussing the procedure, the doctor can either discuss the success rate or the failure rate. And in both cases, the facts being presented are obviously the same, but there is a difference in how it's framed and therefore in how the patient will react. And in this particular case, studies have shown that consent is higher when success rate is presented rather than failure rate, even though, again, they're providing the same information. So in this case, the positive framing is more successful. Another example that's a little different is how to present the choice of vaccinating your child, either that vaccinations protect your child against disease or that not vaccinating your child fails to protect them against disease. And in this case, studies have actually shown that it's the negative framing, so not vaccinating your kids fails to protect your child against disease, that results in higher vaccination rates. So framing can really matter. Another thing that really matters is what your tweets look like. So specifically, it has been shown that tweets with images receive an average of 18% more clicks and 150% more retweets, which makes sense because obviously an image makes a tweet more interesting and it's more likely that you're going to stop and read the content. So I've got a few examples here. So you can see from this top tweet, it has no image. It's just a block of text, not very engaging, and you can see it only received a single like. Below it, we have a tweet from Nature News. It's a little bit better. It's got this sort of thumbnail image of a figure from the paper, but you can't really see it super well. It performed a little better, 4 likes and 3 retweets. But over on the right, we have a tweet from CCRM with this really beautiful microscopy image of a cell. Super engaging. It received 15 likes and 9 retweets. So it performed much better. So images really do matter. And another way to increase engagement is to use targeted hashtags. So For those of you who may not know, hashtags are tags that allow users to easily find messages with a specific theme or content. And on average, tweets with hashtags do receive 2 times more engagement than tweets without hashtags. So when we're talking about science communication, there are 3 main categories of hashtags that you might want to consider. The first is a topic hashtag. So this can include things like hashtag gene therapy, HIV, or organoids. So these are hashtags that someone might search on Twitter if they want to find tweets related to any of these topics. Community hashtags relate to particular groups of people. And a couple of science examples include hashtag SciComm, Women in STEM, or Science Twitter. Again, researchers do search these hashtags if they're trying to find relevant content. Lastly, and perhaps I think most interestingly, there are also conference hashtags. Now, I didn't look in depth, so I don't know if there is a hashtag for this conference, but they do exist for many scientific conferences such as SSSCR or Tillamoocullough. They usually have a unique hashtag for each year. And these are really great because they can be used to share what's being presented at the conference. So as an example, Stem Cell attended the AAAS conference in Washington, DC a few years ago, and they hosted a panel and they live tweeted about it. So what I mean by this is that every single time that a presenter went up to speak, a photo was posted of that presenter, including an introduction of who they were. And then in addition, all the points they were discussing were tweeted, including some additional resources and images that would help with the discussion. And then at the very end, all of the audience questions were tweeted, as well as the responses from the panelists. And because each tweet used the conference hashtag and they were linked together as a Twitter thread, it made it super easy for anyone not in attendance at the meeting to still follow along. So of course, in this case, it was Stem Cell that did the tweeting, but this is not limited to companies. It really is increasingly being done by researchers, especially those who are passionate about science communication, really making the content from these conferences accessible to everyone. And so because of this, some conferences and even poster sessions are actually starting to move entirely over to Twitter. So a Twitter conference or poster session is a virtual conference or poster session that takes place entirely on Twitter using a hashtag. So these conferences work very much like a regular meeting. There will generally be a number of speakers whose talks will be scheduled over a certain time period. And as an example, I have linked to one of these presentations here, which I'll open up. So as you can see here, the organizers have come in and they've introduced the speaker. And then in a thread underneath, the speaker comes in. And in the case of this conference, the speaker had 10 tweets to get across their talk, which might not seem like a lot given that 280 character limit. But again, consider that you can also include figures. You can link to papers so that people can read additional information. So again, very much like a regular talk. And then at the very end, again, people are able to come in and they're able to ask questions to the speaker and the speaker answers. So again, a great way to make things more accessible. And the poster sessions work very similarly. So I've also got one here that I'll pull up. So you can see here in the first tweet, someone posts their poster. It's literally the same type of poster that they would have gotten printed off and brought to a conference. And then underneath, they go and do a little introduction to it. And then they actually blow up the figures, talk about them one by one, and people can come in, including judges, ask them questions, and they can respond. So again, just a really, really great way to make science more accessible. Now, the very last thing that you can do to increase your engagement with tweets is to mention and tag others. And the reason for this is that when a user is tagged on Twitter with the @ symbol, that user is notified and they're more likely to interact with and share the tweet. So when it comes to science communication, I always recommend tagging any researchers whose research you might be discussing, tagging institutions or organizations related to the research, tagging a journal if you're discussing a specific paper, and also tagging policymakers if your goal is to affect change at the government level. It's just a really, really easy way to get your tweets in front of people who can really help amplify your content. And lastly, don't forget to have fun. So you can find a lot of really great and amusing science content on Twitter as well. I've included a few examples here. So on the left-hand side, this is an account from a friend of mine from grad school. So he started the Chromatin Haiku account. And on this account, he does exactly as the name suggests. He reads chromatin papers, and then he summarizes them in haiku form. And he's become incredibly popular with thousands of followers. And he's often even retweeted by some of the big scientific journals like Nature and Cell, which I do think is pretty cool. There's also a lot of really fun science memes that crop up from time to time on Twitter. So last year, #unscienceananimal took off. So this was where researchers, mostly ecologists, would post a picture of the organism they studied and they would unscience it for the general public, often in hilarious ways. Or in the third case here, we have a meme relating to DNA crossover events and the Marvel Cinematic Universe, which I am a fan of. So just to go back to the very beginning of the presentation, we did discuss those traditional channels of science communication that scientists use to share their research, and we touched on their limitation of catering predominantly to scientists. So I'm hoping that after today, I convinced you that communicating your research to the public is important and that Twitter in particular is a great platform for doing so. So thank you for listening, and I'm happy to take any questions that you might have.
Thank you so much, Dr. McBurney. That was an excellent presentation. And being a little adverse to social media myself, I think it was an important lesson for me. I'm looking at the time, and I think it would be best if we moved on to the panel discussion, which you are also a member of. So I'm sure our attendees can ask you any questions there itself. So this brings us to the end of our formal presentations in the Laws and Business Hall. Thank you once again, Dr. McBurney, and our attendees for being here. For now, we will redesignate you, Dr. McBurney, to attendee status, but we will see you shortly at the panel discussion. So in a couple of minutes, we will be hosting a lively panel discussion featuring Laws and Business Hall speakers, Dr. Luc Vandenberg and Dr. Christina Mcburney, as well as a new speaker, Dr. Amy Zarzetsny. I would also like to encourage our attendees to rotate around the research and bioethics halls for the exciting panels there. So with that, I think we can get started with the Laws and Business panel. Let me just check if all our speakers are here. And yes, they are. It's excellent. So welcome, everyone. My name is Bipasha, and I will be your moderator for this panel along with Nicole. We are extremely lucky to have with us three amazing panelists who we will now promote to speaker status. So please be prepared to share your audio and video. And while we're getting that done, I'll start off with the introductions to all our panelists. First off, we have Dr. Luc Vandenberg, who was also the first speaker in the hall and presented on the topic, the academic industry interface, lessons learned in gene therapy. Dr. Luc Vandenberg is an associate prof in the Department of Ophthalmology at Harvard University, an associate member of the Broad Institute of Harvard and MIT, and the director of the Grausbeck Gene Therapy Center. Dr. Vandenberg's work focuses on gene therapy and the study of the fundamental mechanisms of gene transfer. Next, we have Dr. Christina Mcburney, who I very recently introduced, but just to remind everyone, she was also the third speaker in this hall and presented on the topic, Science Communication and Social Media, More Than Just a Hashtag. Dr. Mcburney is the manager of scientific communications at the biotechnology company Stem Cell Technologies, and her work primarily focuses on creating quality educational and news content for those in life science fields. And last but not least, please join me in welcoming Dr. Amy Zarzetsny. Please correct me